Those are the AP guys.
And then if you
Probably recap our things on our funds.
We have it on paper.
Controller.
It's funny. Morning.
You guys won't eat too much.
Oh, that's fine.
You'll just help me get to the U.S.
So John, what you see on the screen and what they see on the outside are perfect. So.
Oh, perfect. That's tremendous.
It's just the people, and we all have this, so it should be fine actually.
Yeah, I can do whatever you want.
Yeah.
It'll be easy.
Just have to imagine.
Or explain it. We explain the whole like support levels, team.
Yeah, we have color shots of all these, so yeah, we can do this normally.
I'm always open for a new challenge, you know.
Yeah. We always put you to the challenge.
Yeah.
Good news on the outside, I can see a perfect thing. We have this.
I'm not going to touch any buttons up here.
There must be some fifth generation quinolone that would fix that.
Oh no.
Just blow that right off the screen.
Yeah, ever. Never cold.
So we're all made.
Yeah.
It's
Calm down. Maybe just
first aid.
If it's in their first aid room, you don't have to go down the hall to get it. See, Al can hook you up with the guys in Snoqualmie. Have you guys ever done that?
No, what's
Yeah.
Just a switch in there. So Al does it in Snoqualmie, and I just.
So you already take first aid and all that.
should be going to a social.
You're basically, the ski patrol's going to do it when you're out. So when you're there, you can give them pain medicine mostly is all they ever want to. And every once in a while we'll relocate and dislocate a shoulder and stuff like that. But you're just there as to answer questions for the ski patrol.
It'd be like a weekend on.
It's like it's a high school boy
I think it's 7 weekends.
It's just like
7 days or something.
Oh, yeah.
sure, whatever. Right. And he's like,
Responsibility.
Today's things like
I'm sure she would.
folks like antibiotics.
oh, it's called snowball. Okay.
You remember, I think we got 2 puppies.
Yeah, no.
Yeah, I know.
That's funny in my head. She did know.
I give these 2 talks in the summer for this season.
I would have done that.
I know. I just can't have the best.
I just wish I had no talking.
I know. See, this is like the older brother, the older brother. More stuff. I know because guess what?
It's like that.
Anyway.
Also, that funny that you're like, weird company.
Yeah. Well, that's the older brother didn't do anything.
Yeah, my didn't pay him for it.
Oh, the older brother, much shyer, much more observer of life.
That was lucky in high school.
Where's my like, can we have a party in our household? I'm like, oh,
So Saturday. Sorry, I'm out of town that day.
and it's the one that we thought was wild. Yeah.
What's the word? So we have it on the you don't see that?
Evidently, yeah.
All right, so you can talk around that. You can see.
I can see
it
yes.
on this screen.
I can see it on that screen. We can make do, I guess.
You should make the word connection is lost.
Which road again?
more, yeah.
Okay, well, let's go ahead and get started. For those who want to claim CME credit, word of the day is microbial. I talked to Drew just last week, and I reminded him that when John spoke a year ago, we had a total technical meltdown. We couldn't project it as far as I remember. He had to talk off a small computer screen that we placed in front of us here. So he assured me there'd be no technical problem this year. However, we can't get this box off the center of the screen here. No one knows how to make that go away. But those of you on the outside, I'm told, don't see it. That you guys see a clean screen. And he sees a clean screen on the computer here. We have the whole thing on paper in front of us. So this time, I think we'll have a good presentation. I know we have a good scientific presentation without, except for the technical issues. So let's go ahead and get started.
Thanks very much. This one. What was that? Okay. Well, thank you very much for bearing with me. Thank you for inviting me back. This is my 4th year here, so yeah, I'll leave it at that. Yeah, 2 technical issues. I missed one because I slept through it, and so batting, I think 1 out of 4 so far. So actually, I'm picking up a little bit where I left off last year. As I've said ever since the first time I've done this group is one of the most fun groups to talk to because you guys are interested in pretty much everything I have to say, or at least what I have to talk about. So I really enjoyed coming here. So I'll go ahead and get started because I think I got a lot of info. I would say the one thing I noticed this year is what I try to do every year is I go through as much of the literature as I can looking for sort of new topics that are overlapping between allergy, immunology, and infectious disease. And at the basic science level, I'm sure there's tons, but I'm not a basic scientist. There's a little bit less of that. So today's a little bit more infectious disease update because this group seems to really enjoy talking about those things as well. I cut out some of the slides that you'll see in your handout. As I was going through this last night, that's when I sort of get more inspiration as opposed to last week. I'm very bad about doing that. So things are trimmed up a little bit. We'll go through it as we can. So as usual, disclosures, I have none, no holdings, no stock, really no pay. So let's start with influenza. So I think influenza is a pretty hot topic over forever, and including this most recent year. This is just the Seattle King County. You can't see it up there at all, but you can look on your slides. This is a cut from the Seattle King County Public Health, looking at where we are. And I can't point to your paper, obviously, but I don't know if you can't see there either. But there's a little black line going up where there's a red line, and the black line is where the weekly percent of ED visits for influenza-like illness is going on right now. So you can see that last year we had a pretty brisk influenza year compared to prior years, and also a bit earlier than typical. Here in Seattle area, it's generally February, March, April, and we're starting to see them in the last couple of years much earlier. We're on track this year as well. The other interesting thing about this year is it looks like it's almost all 2009 H1N1, the PDM 2009. That's the same big 2009 pandemic virus. It seemed to affect a lot of young people, pregnant folks, and that's what's coming through again now. And just sort of on the street experience appears to show that we're still seeing a lot of young people getting infected this year. Plenty of folks who are older and at risk, but still lots of young folks, just like that 2009 outbreak we had before. So we'll see where that black trend line sort of goes over the next few months. One of the other interesting things when you think about where our flu season is that, historically a lot of that flu season was based on diagnostics that weren't very good. The flu antigen tests, the nasal washes and stuff, which are terrible to collect, not very good tests, low sensitivity and specificity, are really being replaced by PCR testing. And as PCR testing is more sensitive and people are starting to test more and more people earlier on, people coming with COPD exacerbations, asthma exacerbations, sort of more things that are atypical for our classic flu presentations, we're probably picking up more cases earlier. And so there's probably some testing bias as well that's driving us towards an early flu season. But you can still see, compared to the big 2009, the green and blue lines on your graphics, we're still not nearly there. This is just a graphic, and it's a little bit small, I'm sorry, but just to give you a sense of the speed with which this is rising. This is the UDub Clinical Virology Lab, and you can just Google this and go to this PDF. It's probably impossible for you to see on your paper, But for those who are looking on the video, you can see that the yellow is influenza A. As I said, that's all 2009 H1N1 now. And you can see it just took off in the beginning of December, just on fire. But interesting as well is that about half of our testing now at the UDub is also positive for RSV. So a lot of respiratory syncytial virus in adults, which historically wasn't such a big, at least thought to be a big viral pathogen in adults, mostly in kids. The current testing in the UDub system tests for influenza A, influenza B, and RSV starting in December. And that's probably some of that testing bias. But we're starting to see about half of our cases sent for influenza testing, even though they're not necessarily looking for RSV, they're getting tested for RSV, and about half of them have RSV, which is sort of interesting development when you think about these novel, what we think of as novel viruses in adults, human metapneumovirus and RSV causing a lot of disease. If I'm going too fast for anyone, please let me know. I know we're going to have a pause here at some point when the AV person shows up. So the other place you can look to sort of see where these things go, I'm not sure if I don't think I talked for this last year, but you can go to Google. You can go to Google for everything else, and this is a really interesting thing. It's called Google Flu Trends. And a few years ago, Google actually started tracking and trending searches for influenza, and then looked at whether that matched with actual flu outcomes, like a flu prevalence in the community. And so what you can see here, this is right now from yesterday. Again, I can't point to it for the folks in the room, but you can see that there's that dark blue line is 2013, 2014. And you can see right at the beginning of December, you start to see a ramp up. And these are searches, people searching for influenza around the world. And what we've been able to show is that you can see that overlaps what I just showed you, those two trend lines in King County and in our testing lab. And you can actually separate this out by city. I separated it out here in that map in the lower right corner by state, and with the darker orange being higher influenza activity. And so right now, Washington State's in a high influenza activity simply based on Google searches. And what's really neat is that when they've actually plotted this, going back to 2004, so the blue is the search trend, in the red, and the orange is the United States actual flu data. And you can look at it practically perfectly overlaps. So this is actually a pretty amazing technology. You can look at simple search trends in a community, trends very nicely, almost perfectly overlapping with actual influenza activity in a community. And yeah, go ahead.
So how do they suss out stomach flu, which happens about the same time as influenza? To most users, that's kind of the same thing, isn't it?
And I think actually clinically, it's often the same thing. So we're seeing a lot of times, particularly in kids, stomach flu is influenza. So the presentation of influenza in children's gastroenteritis, very, very common. And so you may be thinking about like winter vomiting disease or norovirus, which happens a lot more in the winter. But a lot of people actually this year, including, are presenting with GI symptoms with their influenza. So I don't know if they, in their algorithm, whether they separate out people saying stomach flu or, I actually don't know what their exact algorithm, and I'm not sure. I think it is available online, but it may be above my station. And what's really, the other thing that's cool about this is that people are searching on Google worldwide, and you can actually go to Google Earth and look at any place on the planet and look at their search terms going on right now. And you can sort of figure out where their influenza activity is going simply based on those search terms, which is a big deal because not every place has access to testing, right? And there's obviously variable access to the internet as well. But testing is probably even harder to get to than the internet these days if you go to much of the world, right? They're not doing PCRs in Western Kenya, but you can get on the internet there and start looking for things. So one of the big issues that came out this year in a big paper in the summer was about influenza vaccination effectiveness. So it was a complex paper looking at whether influenza vaccination actually protected folks. And I'm not presenting the data from that, but roughly the estimate was it's between 60 and 65, 64% effective in the community. This effectiveness goes down greater than 65 years of age and obviously less than 6 months. So, but for most folks who are getting the vaccination, it didn't prove to be like a super effective vaccine. Now, the idea is that the more people get vaccinated, you sort of develop better immunity across the population. Unlike hepatitis B, it's obviously not sterilizing immunity, but vaccination. But if you get more people vaccinated, you're going to decrease the burden of the virus in the community. So, not a great vaccine. Very few people in the community actually get it. It's definitely less than 50% coverage in the United States. Given all that, this is a nice paper that came out of MMWR just in December, looking at what actually happened as a result of flu vaccination, even given not great coverage and not a great vaccine, and lots of people in the population it doesn't really work for. And the bullets are down here below. You can't see them here, but I think you can see them on your paper sheet. There's they estimated about nearly 7 million fewer illnesses in the United States, which I think is pretty profound, with about 3 million fewer medically attended illnesses, so people going to urgent care, going to the emergency room, going to the primary care doc, and about 80,000 fewer hospitalizations. So even given this, I think people pretty agree that it's not a great vaccine. The effectiveness of it in actually preventing illness still is quite profound. So it may not prevent influenza acquisition, but it does appear to at least mediate the effects of influenza in those folks who get it. And so we're hoping that folks not only don't show up in the hospital, don't show up in their primary care doctor's office, but they just don't get as sick.
Is this typical for all the flu vaccines, these statistics, or just the H1N1?
No. So right now, this is based on the 3-part vaccine, so the 2 A's and single B vaccination. We've used the same one actually for the last 3 years. There's been a change in the B this year. So the A's, though, which is mostly what's circulating community, this is talking about those.
The current vaccine has the right strain in it for what's showing up in the community?
Well, yes. I mean, there's some caveats, and I think that there's the reason you have to get vaccinated every year is, one, your immunity tends to wane over time, even for a healthy young adult. It's probably 10 to 12 months. You start seeing waning, but there's also some subtle drift in the flu A viruses that occur over the year that probably you need to get boosted up against, or basically revaccinated against at that point, which I don't think we have a good handle on that level of subtlety in that drift over the year. Yeah, so this is a standard one.
Then the nasal spray?
Yes.
That's supposed to be more effective than the injection?
Yeah, slightly more effective, in the population who can get it. So right now I think it's still limited to folks 50 and younger. It is a live, it's attenuated virus, and it appears to be slightly better.
And then with asthmatics, at least, we don't tend to give it to them.
Yeah, because you're inhaling it.
Yeah, but
it may be better. But I'll tell you, I'll show you a couple things here. So what's the future of the flu vaccine? So as of this year, we have our first quadrivalent vaccine. So the burden of influenza B virus is probably much greater than we've thought. As I said, our testing is getting better and better. We don't subtype influenza B in our system, and most systems don't. Even with the PCR, we just say influenza B, yes or no. We don't say whether it's the Yamagata strain or the other influenza B strains. And so this year, the first IM, killed vaccine came out that includes both circulating A's and both B's. There's really only 2 B's that circulate out in the world right now. And so that's the first quadrivalent vaccine. And I think that this is probably going to be what's going to happen in the future. If, a sizable proportion of your influenza illness in the community is due to B, and we just miss it, right? So basically a flip of the coin. Which of the B is going to come around? Sometimes we're wrong. If you just include B, you may be able to get that coverage in automatically now. Stop worrying about it. The other thing is probably useful for this group is we have now 2 egg-free influenza virus vaccines, which is fantastic because especially in healthcare workers, there seems to be a very large amount of reported egg allergies. So 2 years ago, the CDC downgraded their warnings. Before that was if you had any egg allergy, no influenza vaccination. Last year they said if you're able to eat things that contain egg, if you didn't have an anaphylactic reaction to egg, you can get vaccinated. We rolled that out, and now people don't have to worry at all. There's 2 egg-free preparations, which is great because that makes people very nervous. And a lot of people report egg allergies without actually ever having been tested or seen an allergist or anything. Sometimes in healthcare workers especially, it's just an excuse to not get vaccinated.
And they eat egg McMuffins on their way to the office.
What's that?
And they eat egg McMuffins on the way to your office.
Yeah, exactly. Yeah, and so it is interesting that the reported egg allergies in healthcare workers is roughly 2 to 3 times greater than the general population. And that's been documented multiple times.
Are there enough variations of the B vaccine? You said there's only really 2.
There's only really 2, yeah. What's that?
Could you just include them in every vaccine?
So I think that's the quadrivalent. Yeah, so I think starting next year, we're going to be probably using quadrivalent, not exclusively, but probably for the most part for our standard IM injection, the UDub system. And I think this is the trend going forward. I think quadrivalent is going to be standard in the future because influenza A circulates in animals and in humans. Influenza B is only a human pathogen, so you don't see the big shifts in influenza B that you see in influenza A. There's no, H3N1, all this stuff only applies to influenza A. Influenza B is pretty just only it has its genetic drift through time. So and there's only those two big circulating strains. So you just plug those both into any vaccine, and you should be good to go.
So why does that have a seasonality then? If it just has humans as a reservoir?
Mm-hmm. It's probably the same things. It's winter. Remember in tropical regions, influenza circulates year-round, just probably not at big epidemic levels. In the northern hemisphere, which has a much higher burden of influenza infection, it probably has to do with inside clustering. But as the PCR testing is become more frequent, people are starting to think about this. And I'd say even in the the 4 years I've been on faculty at Harborview, we've gone from saying this is a winter disease to people getting tested and seeing cases through the summer, especially influenza B. So people get tested all year, and you see cases that we used to never test people during the summer. And now people are testing because you can get the test done, and it's easy, and it's accurate, and we're seeing cases. So we've gone from being a big spike, basically because that's when we test, to testing more frequently during the year and seeing trickles of influenza constantly.
There is also a better adhesion of the virus to the epithelium in a lower temperature. So it was shown.
There we go. I'm not a basic scientist anymore. And then there's two other things I want to just touch base on. One is use of adjuvants. So I'll describe one vaccine. H5N1, the avian influenza, now has there is an adjuvanted vaccine. Adjuvants clearly boost the immune response, and people are working hard on how to get that kind of poorly effective, sort of that 60-65% influenza vaccine to be much better. And one way about that is to add an adjuvant to it. And there's many different adjuvants. There's people who spend their entire career working on nothing but adjuvants. They have pluses and minuses. There's the adjuvant is being used in this H5N1 when it was using another vaccine, had a high incidence of narcolepsy associated with it. So you have to be a little careful, but most adjuvants are quite safe, and that's probably going to be something that's going to be coming out in the future. And this idea of a universal vaccine. So one of the problems with the vaccines we use now is it's based on these drifting and shifting epitopes, these antigens in the vaccine in the virus that change constantly. And the idea is can we get something that's more conserved that can be used all the time, so you can get the same vaccine once, like you do for another virus like hepatitis A or B, and you have more persisting immunity over time, not having to worry about that. So these are places of active investigation. I couldn't find a slide for this, so I drew it. So, so H5N1, just a quick update. So this is the famous bird flu. It's a zoonotic flu. As I mentioned, influenza B is a human virus. It just drifts through time. H5N1 is a zoonotic influenza, and all the A's are. And people have talked about this. I'm sure you've heard about it. I'll show you just some of the numbers on it. This is the avian flu, bird flu, in probably wild birds and in domesticated birds. And people, just so you know why it's one, at least idea as to why, and we may have an expert in the audience here in the corner who can correct me, but one of the ideas is why it's so bad and why it's so poorly transmissible is that it appears to bind to this kind of the normal receptors influenza does, but the specific sialic galactose that are deeper in the respiratory system. So you have to get a good dose of it and get it deep into the respiratory system where it binds to these receptors. And they tend to be, because they're deeper in there, can cause more inflammation and disease, pneumonia-like disease, respiratory disease, compared to standard influenza, which can bind to receptors in the upper airway and the lower airway. And because the standard influenza A's that we see, they're more easily bind upper airway, they can be more easily transmissible because they sneezed and coughed and put on your hands and carry around. Whereas the H5N1, because it's so deep and not binding the upper airway receptors, you don't get the easy transmissibility, which is fortunate. So when you look at this, since it was first diagnosed or discovered in 1997, there's, as of October 2013, been about 640 known infections. Now, that's probably an underestimate because a lot of people aren't getting tested for this. But there's, of the known infections, about half of them have died. There is, as I mentioned, this H5N1 vaccine out there. It's an adjuvant vaccine. This just got approved in the last couple of months in Europe. I don't know if any where it is in terms of the FDA right now. But you can see sort of over time, and again, I can't point to it too much here, but I would say that there's these sort of spikes of cases. In general, I think there's a trend downward from the, over the last 10 years or so, which is ultimately good. One of the things is we spend a lot of attention on viruses like H5N1, but the absolute number of cases compared to the number of cases of standard influenza A in the world, it's a tiny, tiny proportion. But I think people just pay attention to it because you can get these rapid shifts in influenza A, recombination events where the H5N1 becomes something new in a very short amount of time. So it's important to pay attention to trends, but it's definitely not an epidemic that's about to reach our shores. Similarly, H7N9, so this first case actually was described since I last saw you in February 2013 with 3 deaths out of 4 cases, and they had a peak in April of last year. So this was interesting because it was discovered in these few cases, and then it was discovered in many places in Eastern China, very rapidly. And they had a total of it's now up a little bit higher than 140 cases and about 45 deaths. So one third of the folks who were diagnosed, and again, probably when you see early epidemics, you always have higher death rates because you're testing people who are sicker compared to later in an epidemic. Still a fairly lethal virus, but what was really amazing was the description across a huge area. This wasn't like a few spots in China that just kept going. It was multiple places at the same time, all within a year across China. And if you look at the literature now, what it really appears is that this is a virus that's found, I wouldn't say commonly, but is found reliably in the live market system. So if you look at bird markets where people go to obtain a live bird for breakfast, lunch, or dinner, that's where you're finding this. And it's spread throughout the population. And again, this is an influenza A. It's an avian influenza. Like all the influenzas A's, they're all zoonotic. They can all move between pigs, birds, and humans pretty easily and just recombine and move through. So again, it's not something like the 2009 H1N1. It's way behind that. Still very few people. We shouldn't be spending huge amounts of resources on this or time, but you can see how these things happen. And I think when I was putting these slides together like a week or two ago, it was the first description of an I figure it was an H8N10 death in a woman. Oh, I forgot to mention that H5N1, just to give you a sense of how fast these things can move. We had a woman who just died from H5N1 in Canada. She flew from Beijing. She's from Canada, flew to Beijing, stayed there for a week or two, was in Vancouver for a layover on her way to, I think, Alberta. And when she got to Alberta, was taken off the plane, went to the hospital, and she died from H5N1. And so that was the first case of H5N1 in the United States. And you can see how sick the person was. She died quite quickly. But that person was sitting in the Vancouver airport for a layover for 4 hours, just 2.5 hours from here. Now, fortunately, those H5N1 doesn't appear to transmit between humans, but these viruses move around. We have a lot of mobility in this world, and you never know what's coming. And I think that's why people pay very close attention to things like H7N9, H5N1, and obviously the pandemic strains we have right now. Just to update people on MERSCoV, this is the Middle East Respiratory Syndrome coronavirus. Remember, SARS was a coronavirus as well. I have a map here that I think you can see. Really focused on the largest number of cases in the Middle East: Saudi Arabia, Jordan, United Arab Emirates, Qatar. I think there's now been a case in Yemen as well. And the cases you see in France and the UK are really folks who had exposures in the Middle East and then went to France or the UK, not new cases developing there. What was interesting about this was that a lot of the cases actually were healthcare exposed. So one of the things I work as in infection control, And a lot of the cases in these were transmission events in healthcare workers, or the patient would get hospitalized in healthcare workers, and then also family transmission. So clear human-to-human transmission, but still not tons of cases. You can see there's only about 145, 140 to 157, 19 probable, but spread pretty quickly, 12 countries in a very short amount of time, and about a third of folks died with this infection. The folks who initially died also had comorbidities. The R naught, the basic reproductive number, was estimated between 0.8 and 1.3. That's basically, for those not up on their R naughts epidemiology, is basically the number of folks who can get infected from one person. So if you have an R naught of 1, that means that if I'm infected, I can infect one more person, basically. A really bad, something like measles has an R naught of 7 or 8. I can infect 7 or 8 other people. An R naught less than 1 doesn't lend itself towards pandemics or epidemics, because that means I'm infecting less than a person per my infection, basically. Now, that depends on the immunity in the population and all those sorts of things. But it's estimated between 0.8 and 1.3. The SARS virus, which spread quite quickly through much of the world, had an R naught at about 0.8. So, definitely a potential for spread, but not necessarily pandemic, lethal sort of spread. And up until probably last week, the reservoir was unknown. And then, actually a couple weeks ago, I guess, there was some concern, you probably heard this in the papers, of camels maybe being the reservoir for this virus. And they had found antibodies, but never the virus, until this report came out last week. Hi. You know how to do this? Oh, fantastic. So we have the AV person for those folks who are online. So they found antibodies, but they didn't actually find the virus until December, or at least a report in December, where they actually found the MERSCoV virus in camels. And clearly there have been epidemiological associations between the camel exposure and the infections. So it sort of makes sense. We're still waiting to hear more confirmation about this. Tell me if I need to do anything. Okay. So we're still waiting for further confirmation, but it appears that this is a virus that's been circulating in camels, potentially, in exposures, particularly men who are working with camels in farms and other agricultural settings. So the other thing about, have anybody seen a camel or hung around a camel? I don't know. You know what they do? What do camels do? They spit. They're mean animals, and they spit. And so you can think about a better respiratory vector than a camel. A spitting animal.
Have other viruses been associated with camel exposures in the past?
Not that I know of, no. So this would be the first time. So we always think about vectors as mosquitoes and nowadays ducks and birds, and now gotta watch out for camels too. And it's a good reminder that pretty much all infectious diseases are zoonotic in origin. I mean, at some point they were in another animal, and that's where we're continuing to see a lot of these things arise. If it wasn't for our interactions with animals, we probably would have very few emerging infectious diseases. Yes?
How many known, have all the known combinations of H and N been described so far since we started tracking the flu?
Yeah, so if I talked to you last year, see if I get the numbers. If anyone knows better, remind me. So no, because there's always new ones. So if I told you last year how many A's, so how many H's and N's, it was probably something like 4 different H's and 3 or 4 or 5 different N's, so the H, N, that were ever seen, sort of known in humans. And this year I would say I think it's 5 or 6 H's and 5 or 6 N's now. So for instance, we never had a 10 before. Now there's a woman who died in China with a H10. The 7, 9, those weren't ever described in humans either. So I would say that probably every year we're seeing new hemagglutinins and new neuraminidase genes, at least within that naming categories, every year.
Okay, so it's still an open-ended infection. There's just new changes all the time.
Right, but not necessarily the big pandemic ones or the ones we see. The influenza A that we vaccinated against is still the H3N2 and the H1N9 2009, the PDM1. So those are the two predominant ones we see. But there's a lot of potential combinations that can potentially go on. And that's why people are so worried, because these influenza A has this predilection to big genetic shifts, where you can get that H1 can be recombined with something else. And you got to remember the big Spanish one was an H1N1 too. And that affected lots of young people, just like this H1N1, even though they're different viruses. The intermediate ones, the 57, or the 57 and the 60s, were both H3, and they're different. They're not H1N1s, and they appear to affect older folks much more consistently.
And the people who die, have they got different receptors for the flu virus, more dense or more?
The 2009, the current flu. No, I think that probably has to do with antibody responses. So I think older folks have some immunological memory from prior exposures that are protecting them, and younger folks don't. But I don't know that. I don't think there's any greater receptor density in those people, because it really seems to be associated with the type of flu as opposed to, the subtype, the H1N1 versus influenza A. But there may be subtleties that I don't know about.
I know a couple of years ago when sort of bioterrorism upticked, as it usually does every year or two, there was a lot of talk about maybe developing a vaccine against more conserved parts of influenza that don't change, which would be logical. Do you know if that's any?
Yeah, so that's the universal vaccine that people are talking about. So definitely, so people want to move away from these hemoglutins and neuraminidase that we know shift and drift, and move towards more conserved epitopes, so that you don't have to get a different vaccine every year. You can get either the same vaccine every year or one vaccine that lasts much longer. Absolutely. So I think the goal would be like an adjuvanted, universal, quadrivalent vaccine that covers, 99% of the strains that are circulating in humans.
Is there a reason there seems to be more shift? Is it just that we're better at testing for more H's and N's now? It's not that we're driving it by putting pigs and chickens together, or?
Well, I think that it's probably both. I think mostly it's testing. So, I think SARS changed everything, especially for China, where we see a lot of the origination for whatever cultural, agricultural factors, the combination of animals and humans in that area seems to be a place that a lot of these viruses pop up. With a caveat that we were all looking at Asia for the next big pandemic strain. In 2009, H1N1 came out of Mexico. No one was paying attention at all, right? It already made it across borders before anyone looked at it, any idea. So but SARS really woke up I think the public health folks worldwide, but also in China. And they were sort of closed-lipped about that initial outbreak and opened up afterwards. And Margaret Chan, who was the head of the WHO, sort of openness, transparency has increased dramatically. And so we're starting to see lots of reports. They realize it's not bad to say these things. It's good, ultimately, for the community.
Why don't you go ahead? I don't have any confidence they're going to fix it. Ever.
I'm going to quickly just do a couple slides about hepatitis C. This is sort of one of these occult epidemics that a lot of folks don't pay a ton of attention to unless you're involved in it directly. So this is just a slide looking at sort of some of the prevalences. So if you look at hepatitis B, there's roughly about 1 million people who are infected. About 2/3 don't know they're infected. There's about 3,000 deaths in 2006 related to that. Vaccine preventable. So we have a great vaccine for that. We're going to see that number go down and down and down because everyone's kids are getting vaccinated up front, right? That's fantastic. It's transmitted by a couple of different ways. Hep C, or I'll just go drop down to HIV, there's about 1 million infections in the United States. About 1/5 of them don't know they're infected. It's about 14,000 deaths per year. That's pretty constant. No vaccine. And you can see it takes up about 70% of the CDC budget in terms of research and control. So move up to the middle one, Hep C. We've got about up to, estimates vary between 3 and 4 million infections. And even worse than hepatitis B, about 3/4 of them don't know they're infected. There's about 12,000 deaths, so approaching the HIV infection related deaths in the United States. No vaccine, and you can see all 3 of them are transmitted in the exact same ways: blood, birth, and sex. Right, the truths of life. And hepatitis B and C combined have about 2% of the budget compared to the 70% of the HIV epidemic. So you can see that there's an imbalance, at least in the United States, priorities in terms of the number of infections. Hepatitis C, huge. The social issues where most people don't know they're infected. A lot of these folks, when I've spoken and I've done Hep C studies, epidemiology studies, you talk to someone and they may have used intravenous drugs in 1972, and now they have a job, and they may be thinking about retirement, and they have cirrhosis. And it's just they don't, just a bit of youthful dalliance and so forth, leading to bad outcomes. We used to kind of downplay the sex part, but there's emerging data showing that, especially in MSM couples, the transmission rate is actually quite high. So men who have sex with men, the transmission rate of hepatitis C between those couples appears to be very high and very quite reliably as a sexually transmitted disease, which we never thought about before. We used to think it was fine to have sex between discordant couples, and I think that's changing. So here's sort of worldwide prevalence, and it's a little hard to see probably on your handout there, but in the lower left-hand corner you can see there's darker boxes with the prevalence of hepatitis C infection at percentages, with dark green being greater than 3%. And you can see there's a few hot spots, but most importantly, you can see that very large areas of Brazil, other parts of South America, Russia, Pakistan, huge percentage. If you think greater than 3% of your population is infected with hepatitis C, you're talking about huge numbers of folks. The United States and China are about equivalent. A lot of these places we just don't have any data on. We used to think hepatitis B and C was not a problem in Africa. It is clearly a problem in Africa. There's clear whole generations of individuals living in West Africa with hepatitis B and C that's been clearly at least associated or identified with colonial vaccination practices. Where the ruling powers, the UK or other European powers, conducted serial vaccination or medication administration with injections across time. They were trying to do the right thing, but there's age cohorts in Cameroon and other West African countries where huge proportions of 60-year-olds have hepatitis B and or C. And this has been shown in a couple of countries in that area. Interestingly, you can see right in the middle, the world's highest prevalence of hepatitis C is in Egypt. And does anyone know why?
Spitting camels.
What's that? Spitting camels, yes. It's always the camels. So Egypt, through the last 30, 40 years, really had a big effort towards improving their agricultural sector. So they have this huge river, the Nile, lots of water and lots of arid land. So they built irrigation ditches in a lot of areas in Egypt in an effort to do this. And they were very successful getting water out of the Nile into dry areas. There's one animal that loves irrigation ditches. They tend to be stagnant. For any of us who've lived or worked on a farm, irrigation ditches kind of still water a lot of times. And those are snails. Snails are one of the vectors for schistosomiasis. You can treat or do big mass drug treatment programs for schisto prevention. It's an injectable drug. And just like I told you about the colonialists back in West Africa, huge populations, numbers of people in Egypt were treated serially for schistosomiasis, just presumptive treatment. And in doing that with the same needle over and over again, infected huge numbers of folks in Egypt. This is an epidemic, literally an epidemic of hepatitis C in Egypt. It's a huge problem of people presenting with active infection, liver disease, and cirrhosis.
How long ago did that take place?
Pardon?
When did that take place?
I'm trying to remember the years. I think it was in the 70s, but it lasted for years. And there's still issues with their drug supplies, their blood supply too. So, yeah, but it's a huge, huge problem in Egypt right now.
Do folks follow like HCV and HIV co-infection?
Mm-hmm. Yeah, there's actually I'm not an expert in that, and I wasn't going to touch base on it too much, but Hep C HIV co-infection is a huge deal. So we have a clinic at Harborview that's nothing but Hep C HIV co-infection patients. Remember, if you look at the transition routes, they're exactly the same. And so you can have Hep B, Hep C, and HIV from doing the same things. And Hep C disease and HIV feed off each other. So you see people with HIV infection with Hep C, their rates of liver disease is much higher and faster. than you see in people without HIV. So getting control of both viruses is actually super important. If you get HIV under control, your Hep C progression is improved. If you get Hep C under control, your HIV control is better. And so they're definitely beating off each other.
Is that because of effects of HIV on the liver or from the drugs treating the HIV?
No, it appears to be the direct effects of the virus. People with untreated HIV have faster progression.
So how can we do a better job of diagnosing these people? How do you pick them up?
Yes, and so that's a great question, and I don't think I'm going to come back to that in a second. Just to again hammer this home, here's HIV mortality in this top bar right here, just above the white box on your screen, and you can see that's going down and down and down. We're getting good progress, not as much as we want, but definitely good progress. The trend's in the right direction. Hep B's in the bottom, below the white box right there, which is flat, maybe slightly going down. But you can see Hep C deaths in the United States are slowly climbing. And this has to do with the idea that very few people who have Hep C actually know they have Hep C until they present with liver disease, cirrhosis, and other things. And so, quickly, as I just sort of recapitulate some of those things, about 4 million people are antibody positive in the U.S., 1.8% of the population, right? There's about up to 4 million people are chronically infected, and the highest prevalence is in 30 to 54-year-olds. And so this kind of gets to your question. So there's some really good economic data, so modeling data, and as well as basic epidemiology data, implying that pretty much every adult should get tested at least once in their life when they show up to the primary care doctor's office. This was never, we used to say, did you ever use IV drugs? Did you ever snort cocaine? Did you ever get a tattoo? We'll test you. And what most modeling data now, and I think the CDC has come around to this, and others are coming around to it. I've seen some extremely promising modeling data saying that our cost savings on the other end, if we diagnose people early, is profound. And this would support testing every adult at least once in his or her life. Because people do things. And on top of that, when you go back and ask people who have hepatitis C, in this one study I worked on, about a third of people said, I never injected drugs, I never snorted cocaine, I don't have any tattoos. And maybe some of that was not true. Maybe it was just not recall. We didn't ask about sex. We didn't use to ask about sex, and that clearly is a vector. And so, but we miss folks a lot. And a lot of folks have no idea that they're infected because you remember this is a quiescent disease for many decades. Most people don't present with acute infection. They show up with liver disease and cirrhosis for those who go on to develop disease. The other thing is that we have treatments now that are much better. Akin to HIV, treatments up to the past couple of years have been quite toxic, expensive, time-consuming. So historically, and you can't see behind your box here, but it's pegylated interferon. So you used to use just straight-up interferon, frequent infection, frequent injections, and oral ribavirin, which made you feel depressed and like you had influenza all the time. Terrible. Associated with anemia, cytopenias, and so forth. Very difficult to treat disease and bad outcomes. A third of people got what it called sustained virological response. Their viral loads went down. And then, starting last year, we started having these protease inhibitors, telaprevir, boceprevir, still being combined with ribavirin, pegylated interferon. The FDA just approved in December a drug called sofosbuvir, which is a chain terminator for hepatitis C, and that shows extremely promising results in the predominant genotype in the United States. That's genotype 1A, which is the hardest one to treat. It just has the worst response. Genotype 2s and 3s get better, a lot of cures with those, but the cures for genotype 1A tends to be less than 50% in a lot of these prior treatments, especially the interferon and ribavirin treatments.
Now, when you say this is quiescent, these people have normal liver enzymes too. So, a lot of people get screened for their cholesterol with a chemistry profile, and they always get ALT and AST.
Yeah, and so I don't know the exact data. I don't know if anyone's got a good study to show the natural history over time of people who are hepatitis C viral load positive, because I just don't think we've been testing people. Most people who are going to go on to develop, most people who get hepatitis C don't go on to develop disease. Probably 80% of them don't, 20% of them do. And so whether there's some people in there with a AST/ALT in the 80s or 60s for many years, a lot of them have kind of that 80s, 60s, 100s over years, and just the process of remodeling, scarring, and cirrhosis just takes time. as opposed to a big liver insult early on.
But when you say screening all these people, I mean, if they have absolutely normal liver enzymes, does that replace a screen serology
I
or
don't
not?
think so. I think we're still testing. So you can do a hepatitis C antibody. Hepatitis C antibody is positive. You can move on to viral load and genotype testing. So the really promising thing is that I just told you taking interferon ribavirin is quite toxic, associated with lots of side effects, and sofosbuvir appears to have actually some data to prove very good outcomes all by itself. It's an oral drug, and probably this year you're going to start seeing 100% oral regimens with sofosbuvir and ribavirin, or maybe even better drugs by the end of the year. There's a lot of drugs coming in the pipeline for this. I just showed you there's millions and millions of Americans who are infected with this. Worldwide, there's millions, billions more. And so the idea that we could an all-oral, highly tolerable regimen is extremely promising. It looks like it's going to happen the next year, which means testing is even more important because now we have better treatment. The downside to this is cost. So single, 24-week or treatment with sofosbuvir is going to be, guess how much? $20,000. Between $60,000 and $100,000 per person. So balancing the economics of this treatment is going to be very interesting as we go forward. But it'll also be available to less specialized. Most people get treated by even an ID. I don't treat people with Hep C. We have subspecialty hepatitis treatment people who do that almost entirely. But this is going to be able to kind of leak back into maybe straight up ID and maybe even out into primary care in the future. Given the number of people, we don't have enough hepatitis specialists to treat this number of people. It has to get out into the community. Okay.
So, excuse me, on screening then, if you've had a hepatitis B immunization, you have antibodies against B.
Yes.
What are you looking for with hepatitis C?
So Hep C, the difference with hepatitis B immunization, there's a whole bunch of different antibodies. So you can have core antibodies, surface antibodies, right. E antibodies. And so you can actually differentiate between infection. So you can't have an S antibody, surface antibody, unless you're immune. So unless you've cleared. So if you're infected, you'll have an S antigen, surface antigen, and your antibody won't convert. So if you're positive, that means you're either vaccinated or been infected and you've cleared. So that's done. So as long as you have that, you're fine. With Hep C antibody, most people with Hep C antibody, they have no vaccine, so that's easy. So there's no other reason to have it. And most people who have Hep C positive antibodies have the infection, right? Most people don't clear it. And so it's economically worthwhile to go on to the next level of testing, get the viral load. And genotype. But genotype is highly associated with cure rates.
Does it matter what class of antibody, G or M, or do you look for that?
Nope, we don't. Straight up, yeah. That'll only be important in acute cases, which are very uncommon. Most people don't come in. I mean, I've seen a handful of people with acute Hep C. They're almost all 25-year-old females who show up jaundiced.
So most people are clinically well when they acquire the virus.
Yep. Yeah, if you're a male who's over 45 and over 40 or whatever, or over 35, they almost never show up with anything. Yeah, and there's some association between the acuity of the onset and clearance too, because you may be just having a better immune response. But unless you're a 25-year-old female, it's probably not going to happen.
The U.S. Preventative Task Force going to make these screening, a recommendation?
Either they have or they're in the process of doing it. I think it's been a big debate.
I mean, the VA requires you screen both HIV every year and Hep C like every 5 or something.
Yeah.
They teach all PCPs now in residency to screen, but
yeah,
not a recommendation.
And I think I think the VA population is a slightly higher risk group than your standard primary care panel, and that's where the argument is. I'm going to test you for this sexually transmitted IV drug using disease, virus, and people are like, what? Why would I do that?
Well, you still, like with HIV, a lot of states have required written informed consent to test. Mm-hmm. That's still such an absurd rule.
It's a completely made up, a disease that's transmitted in exactly the same way as hepatitis A and hepatitis B and hepatitis C requires some sort of special consent. That's, I think, historically
Well, it was in about half of states. It was still required.
Here, you better document you've got consent on your note, otherwise you could get in trouble. Really? In our state, yeah, you have to. You don't have to have a special consent form, but you have to document that you consented the patient for HIV testing. Hepatitis C and hepatitis B requires no consent whatsoever. You can do it standard as a separate test. But I think most primary care providers probably say, well, we're going to check your cholesterol today. We'll check Hep C. And there's going to have to be a conversation about that. Hopefully it's just going to be a one-time test for the vast majority of people. But we got to get a handle on this huge number of people who are infected, especially if we have potential treatments. So I'm supposed to go to 8 o'clock, right?
Yeah.
Okay. Well, I'm just going to give you the ID update. We'll not talk about antibiotics until next year. Sorry. So here's an interesting case that came out last year. Cured of HIV. So did anybody hear about this? It was in the news. I'm not sure if you guys all know the details. So it's really interesting. So a mother showed up in Mississippi to the hospital, ready to go, ready to give birth. She had no prenatal care. They did a rapid test in her mouth and was HIV positive. And the baby, before they could get the zidovudine in, the baby was born. And typically you treat a baby with the verapine or zidovudine for a period of time, 6 weeks or so, after birth as prophylaxis. What the person who delivered this baby decided to do was just to treat the baby as if she was infected, which is kind of a chancy thing, right? Because you don't have data, you don't have the labs back, you don't know where this baby's infected. But went ahead and did it. She just thought it was a high-risk situation. Mom probably was untreated HIV. You don't know what stage she is. And so the influence started on lamivudine, zidovudine, and nevirapine in 30 hours. Within the first week was changed a little bit to a protease-based regimen. She got the lab samples from the PBMC at 30 and 31 hours, which is the standard testing. And both of those were positive for HIV virus in the baby at 30 and 31 hours. She didn't know that when she started the treatment, right? Those tests didn't come back for a couple days. But the baby clearly had virus in her body, and in her PBMCs and in her blood, so she had a viral load. And this remained detectable, multiple testing over 19 days, with a trend going down and down and down. So the baby continued, so in some ways kind of got lucky, started in this pretty toxic, fetus or little baby infant, started in this pretty toxic regimen. The baby was infected and was continued on treatment. And the repeat viral loads of the first year of life were all undetected. So drugs appeared to be working. And then around 18 months of age, the baby was lost to follow-up. The mom just disappeared. And she showed up at 23 months with the baby. And it appeared that mom said, I stopped giving her the meds when she was 18 months old. The last pharmacy refill had been about 15 months. So the baby had been off meds for months. And the pediatrician sent off the labs, fully expecting the viral load to be sky high. It's a little baby with HIV, they accelerate very fast. And it was undetectable. There was no virus found. And at the time of the publication in the journal last year, they had 30 months of HIV PCRs, the antibody tests, which were all negative, off therapy. The baby never restarted therapy. And in real time, it's been about 2 years. The baby's not had any detectable virus. This samples from this infant have gone on to very high-level, single-level viral copy testing. And there's been some hint of DNA material, HIV-associated DNA material in this baby fluctuating, but never a viral load, never a full virus, and still no antibodies, which we know people with HIV develop profoundly good antibody responses. So, the idea of a cure came up. People have always talked about early treatment, and I should have put that cured in quotes because it's a very hotly debated term. It is probably the wrong term, which is why I put a question mark. What happened with this infant is unknown. And I put over on the left side, you can't really see it because of this other garbage here, but on your paper you can see there's confirmation of the HIV infection. You can see the HIV-1 viral load in black going down to 0. HIV replication over here, you can see the drug dosing. And then the CD4 count, this child has actually better than average CD4 counts for a child of her age. And this is really the first time this has ever been described where someone got treated and stopped their meds and had no virus afterwards. Now, there were trials of these treatment interruptions, 5-10 years ago, which all were associated with higher mortality in people with HIV. So people who got treatment interruptions because of toxicities, their viral loads would go to 0 for some amount of time, then they stop their meds for 2 months or 6 months, and then they wait for the viral loads to climb up and then restart meds, try to give them these little holidays. It's called structured treatment interruption, and that was clearly associated with higher mortality. More people died who did that than if they just stayed on their meds, even with the toxicities. But this has started to make, when people talk about cures, this is what's happening. Can I stop my meds? Can I stop my meds? Maybe I'm like this baby. That's definitely not the situation. This is a very unique circumstance. It helps, it's one of those single cases, gives us some insight into the immune system. And what can potentially happen, this baby was treated very early. What's interesting is when you think about the modes of transmission in infants, vertical transmission, is that babies can be infected in utero, perinatally, so as they're being born and they probably inhale secretions and blood, or via breastfeeding. And probably most kids get infected in either as they're being born or via breastfeeding, as opposed to in utero. This baby had virus while she was, 30 hours old, which would imply she was infected in utero, which is an uncommon way. Well, that would mean that she was infected. She was not, infected just as she passed through. We got it super early. She got infected sometime in those 9 months, probably in the second or third trimester, potentially. And so that would imply that she had percolating virus cooking along, floating around with her PBMC for quite a while. But not as opposed to she's just got infected as she was being given birth, and then we gave her the drugs, and then it all got cured. So I just wanted to throw it out there as kind of an interesting thing that came out last year. Got a lot of press, New England Journal paper.
Has this changed the health of the mother? Is that fair on this in any way? Do we know the mother had a strain that was less severe?
No, I think she had a pretty high viral load.
Has this changed any of the known exposure prophylaxis in people that have a known sex partner, a needle stick injury with a seropositive person?
No, those standards, actually, if anything, those have gotten more aggressive. So just starting this year, we historically were separating people into, no risk or extremely low risk, medium risk, and high risk, basically. And for medium risk, you get 2 antiretroviral drugs. For high risk, you get the full treatment dose of 3 antiretroviral drugs.
Is it these same drugs?
Actually, historically, some of them were. Now, we've actually, because the more tolerable regimens, there's one drug called raltegravir, which is very tolerable. Lamivudine it's called Truvada, so it's lamivudine and is it AZT? Lamivudine is one of them, which is one of the drugs this child's getting. The other 2 aren't. And now basically everyone who has a reasonable exposure gets the 3 drugs, gets treatment doses. There's no more medium risk. It's all one risk. Either no drugs or all 3 every time. It's because historically when you gave people the old drugs, they're pretty hard. Almost no one finished their 28 days because it's just made people feel awful. The drugs are much more tolerable now. Providers who are doing the ER or other place, urgent care, this decision tree between medium was always very troubling. Blood in my eye, blood in my mouth, needle stick, hollow bore, solid bore in the OR going through gut versus, or blood draw. It's just too much of a mess. So now if you're going to treat people, you just treat them with treatment doses of drugs.
Do they know if this child was breastfed or did they?
Not breastfed, which is one of the major ways for transmission.
I was just wondering if she had been breastfed, if it showed any type of protection throughout.
So breastfeeding is complex with transmission. So if you look at an untreated population, and I need to stop soon, but if you look at an untreated population of mothers with HIV, probably 20% transmit via breastfeeding. But depending on and so more kids get infected and do poorly in places like the United States. If you do the same thing in underdeveloped or low resource setting like Ethiopia, where if they don't breastfeed, they have to use formula but don't have clean water. More of the kids who don't breastfeed die because they have more respiratory and gastrointestinal infections. So more pneumonia, gastroenteritis. So it's actually safer in a low resource setting without access to clean water to breastfeed with an HIV positive mother, even if you're uninfected, than the other way around. And this was historically, we used to say, oh no, don't breastfeed. Well, these kids were dying. They'd be drinking dirty water and other things. And so in the United States, if your mom has HIV or is at risk for HIV, then there's just 0 breastfeeding. That's the goal. even if you're on therapy. People who are on therapy have very low transmission risk, though. There's been studies now in discordant couples that people who have suppressed viral loads really don't transmit infection. It's probably scientifically safe to breastfeed, but there's no benefit for HIV immunity, clearly.
I guess I was just wondering if the child on that treatment regimen would have had some type of protection if she had been. I guess
we've looked at it. Actually, it's been studied, and no, it doesn't appear so. It's interesting. I have to stop now, but the antibodies that are transmitted, humans don't. We get antibodies from a mother via breastfeeding, which are very important, which protects us against gastrointestinal respiratory infections. But the antibodies don't transit across the GI tract. Cows do. So you can actually get antibodies in a calf in the blood transmitted from the calf's mother, from the cow. But we don't transit those antibodies. They all stay kind of external in us. We're just big tubes. And so I'm going to stop there. It's always pleasant to answer questions, but I just want to say, so next year,
Something else.
we'll talk about antibiotic resistance, because the CDC came out with this big document this year talking about the antibiotic resistance threats. So antibiotic resistance has gotten to the level of a security threat to the United States. And so people are very active and interested in this. And it's something that I spend a lot of my time working on, and I look forward to talking to you about. So thank you for your attention. I'm happy to stay here as long as you need me to and answer questions.
This is a very broad question from someone who knows virtually nothing about current HIV, but there have been a few other cases of supposed cure of this disease by bone marrow transplant.
So there's one.
One, and also about people who lack the receptor to ever become infected. That may be a very broad question.
No, it's actually, there's the same answer in many ways. So for HIV to bind to a cell surface, it needs two receptors. It needs CD4, right? So CD4 cells, the cells that we know go down, T cells that go down when you're infected. And it needs co-receptor. So the one for initial early infection is called CCR5. It's a chemokine receptor. There's other receptors that can bind, but the ones that are in the mucosa, which is the majority, heterosexual transmission is the biggest transmitter or mode of transmission in the world. So the mucosa are on dendritic cells, macrophages, and they have CCR5 and CD4. So the most viruses that initially infect someone are ones that use CCR5 and CD4. Now, so that means that if you have CCR5, you're set up for infection. So in Europe, in Northern Europe particularly, there's a proportion of people who are CCR5 heterozygous, CCR5 null, heterozygous or homozygous. So there's homozygous folks who have no CCR5. It appears there's no immunological problem with not having CCR5 in these individuals. And those folks appear to be relatively resistant to infection because they don't have that co-receptor at the mucosa. Now, there are people who have CCR5 null who, in some blood transfusions, something like that, who have gone on to develop disease. But in general, it makes them very resistant.
Now the screen's clear.
Hey! Start for the whole year. Start for a whole another hour. I have a whole another hour of slides here, I think. So I'm always over enthusiastic. So what's interesting about the Berlin patient was he was a patient with HIV infection, and he had multiple myeloma or some other liquid tumor. And when he got transplanted in Germany, he's actually from here, Seattle area. When he was in Germany and he developed this cancer, his oncologist kind of smartly said, let's see if we can find a match that's CCR5 null, negative. And he was in Northern Europe, so it made sense. He was able to find someone who matched him, who had no CCR5 or had a CCR5 null phenotype or genotype and phenotype, and transplant him with that. And so basically had all these cells now that were resistant to infection. And he's been HIV negative since then, undetectable viral load. So basically cured with a bone marrow transplant. Bone marrow transplants have been tried in the past and continue to be tried, not necessarily with CCR5 null transplants, but certainly other type of transplants, and it doesn't work. And there's no way we'd be able to transplant everyone with CCR5 null cells. Bone marrow transplant is a pretty deadly disease thing to do to someone. It's very dangerous, and so as a treatment, problematic. But as a case sort of like this one very interesting. Because it's interesting because the longer you're infected, the more diverse the tropism of the virus becomes. So there's other ones, CXCR3, CXCR4, which are other co-receptors. So initial infection is sort of CCR5 using trophic, so it's macrophage trophic, and it becomes T-cell trophic over time, more CXCR4 using. And so the virus is able to use more and more different co-receptors over time. So you think the longer you're infected, the more diverse your population is, or actually more biases towards non-macrophage trophic virus. And so you'd think that late infection like this patient was, you'd have a more heterogeneous population, that CCR5 was, just getting rid of CCR5 would be enough, but it appeared to be for him. So, pretty cool.
There's just a couple papers in Nature, I think a couple weeks ago. It was cast down in San Francisco. They were saying it was a new immunopathogenesis of HIV. It was caspase induction. It was causing apoptosis of the...
well, this is the thing that's been amazing. I've been saying
it might be some new therapeutic approach then.
Yeah, I mean, I think there's always new things. The thing that's been remarkable to me, and I've there's been signals of this in the past. I haven't read those papers, but it is, to me, abundantly clear that HIV entered the human population at an epidemic level, probably in the 50s or earlier. But HIV has clearly is adapted to humans. And I'm willing to bet that HIV has been percolating in the human population for thousands and thousands of years. Because it is remarkably adapted to not only our adaptive immune system, antibodies and T cells, but to our intracellular immune systems, our non-adaptive immune systems. If you look at the signaling machinery within a cell, like caspase induction and so forth, HIV is able to work at that and prevent, single cell apoptosis machinery or induce apoptosis within the cells at a remarkably sophisticated way. And so I'm not at all surprised that we're still early in our understanding of HIV pathogenesis. I mean, no one's ever understood totally why T cells disappear at the rate they do.
I forget what the drug was, but they had a drug that they were for, some totally other random disease versus HIV. It was an epilepsy drug or something, but it blocked the transmission in vitro, so they were going to try to get clinical trial.
Yeah, I mean, I think that there's still much to do about HIV. There's still lots to learn. People are still, right across here, Keith Jerome and his colleagues are still looking at how to snip HIV out of genomes. The cure is not going to be, I don't think, my personal opinion is it's not going to be a vaccine and it's not going to be bone marrow transplant. But it may be something, I think we're going to have to develop something completely novel, like looking at other drugs or other treatments.
I was going to suggest, I've wondered why it's been so hard to make a vaccine. A lot of effort and money has gone into it. I don't know if that's a worthwhile thing to discuss partially next year.
Yeah, I can talk about that. It's, I'm definitely biased. There's probably better people to talk about it than me because of my bias. But I worked in HIV vaccine stuff for 10 years or more in both macaques and humans. And I don't see, the problem is that if I take, I can get people with HIV infection have very powerful T cell responses and very powerful antibody responses, including neutralizing antibodies to infection. And the mutation rate and the ability for the virus to get around each of those barriers, immunological barriers, is profound. It's able to get by, mutate past T cell responses. It's able to get by, mutate past antibody responses. So as soon as you mount a potent neutralizing antibody response, you come back. The virus comes back. And so and in those babies, for instance, if I get vertical transmission of antibodies into a baby via, from birth, so babies who are born have a big chunk of their mom's antibody response, it doesn't protect them at all against HIV infection via breastfeeding. So you have big immune response to via antibodies in an infant who's born, who's uninfected. You breastfeed, they breastfeed, and they still get infected. There's no difference between having a good immune response and not having a good immune response in those kids directly against HIV. So we don't know how to do it, and the diversity of HIV is so enormous that it pales compared to how we deal with influenza vaccination, which requires yearly, and it's still not that good. So you'd have to make a unique vaccine for each individual based on who they're going to be exposed to, which you don't know. And we don't even know that. So there's no shared conserved epitopes that work for prevention. And so it's very tricky. And it's so good at getting by all these systems. It's just, I'm not very hopeful that that's the way to go. Doesn't mean we should stop. I don't think so. We've been successful in other things, but I think you've definitely seen some of the fervor kind of calm down. And there's lots of other things we got to deal with at the same time. So I don't know. But there's people. We have some of the world's best HIV vaccine people here who could definitely give you a different, more global insight to that. I did it for a long time, and I was like, I don't see any promise here.
Well, we tried to screw you up, but you pulled through anyway.
Well, I was probably more optimistic than I should have been, but I appreciate you guys listening to me talk about it. Well, infectious diseases.
If you'll come back next year, we'll have you. Thank you.
Yeah, thank you.
I think it's
Obviously, now it's perfectly correct situation. Something programmed into the No, it's rooted every week, almost every week. Unpredictable.
Yeah, not every Monday, so sometimes.
People Jerry this year. We typically have that UDub.
Oh, Jerry Hamilton?
Yeah,
he wants to have it again.
I don't know whether Drew wants this to talk. I'm just getting rid of the old one.
Oh, yeah, that's probably a good idea.
That's the thing.
This is one. This is the new one here.
Okay. Yeah, just leave it on there.
Are you talking about ART or separate?
Separate. The one,
Yeah, Jerry is planning to do one with Genentech on Sunday.
Of course. Hi. No. Oh, no. Absolutely not.
Trying to find a paper.
But you would think that she'd be. I'm trying to remember. I have to go back and look. That's a good question because most kids are positive for about
open for dinner
6 months or so.
Friday,
Yeah,
Saturday night.
I don't remember exactly. And like I said, luckily it turned out right, and was able to get some people from UMass and Hopkins involved and did a lot of testing. And,
No problem.
And
Okay.
some other stuff.
Yeah,
I love it.
Yeah, how are you?
Oh, you want more
information?
I'm going to try.
Yeah, and it's just like, I don't know, I'm a visual guy. For me, writing it down gets it imprinted, but if I'm...
It's almost like a charade that I can write it down, but I already know that I just remembered it.
Yeah.
But it's hard actually to interact with. So the reason I want to ask or talk to you is I've been talking with King's resident Josh Parliament. He's interested in allergy.
Oh, right.
And Max was here. We only applied in a couple places. And
I was going to.
what's your best advice? I mean, should you talk to him at some point if he's interested in actually seeing the future? Like, how do I counsel him on how to do the right thing? I don't know what to say.
How much longer is he here?
he's graduating now, or end of the year.
In a couple months in June.
June, yeah. And so he's got some time. He's looking for what he wants to do next. But should he come talk to you and sort of get ideas about what?
Yeah.
How to convert?
Yeah, you know, Drew,
okay.
Frank.
Yeah. We only have 2 slots to record, right? And so
yeah, 100 applicants.
100. You know, I think narrow the scope where you apply it, and if you want to say in Seattle.
Yeah, you got to have a wide.
I try to counsel him. I said, you know, I have to go to a state you don't like. Or he doesn't like you.
Where'd he go?
So maybe I'll talk to
He
you.
didn't say. He just said that he had to
Yeah,
leave
he's
a little
looking for
earlier.
So
So he may have gone to the clinic.
Gold points.
Matt said. So
I know, Matt, you know we're not having to do it too early.
I'll let him know.
Yeah,
But you're going to take everything upstairs, Amy?
yeah, I'll take the food that maybe.
By the way, next Tuesday,
I really
you're having cataract surgery Monday.
Oh, this next Tuesday? Okay, yeah.
So I don't know if I can
did
come
some
next
lectures
week.
over at Children's.
I don't know how I'll feel
now.
Tuesday
Oh,
morning. No, I
yeah.
can't drive.
Yeah, right.
Yeah, but I'm more like a legislative
If Matt
guy,
drives
so
me,
that's
I
not.
won't come
I'm
at
going to
all.
be coming in person, so.
Well, that's it. He worked with you.
But I'll let you.
Right, yeah.
Yeah, I have
And so we actually have some more stuff coming this year. We're actually, not me, but the senator's going to drop probably a Good Samaritan bill for Washington State. So that passes. I'm not sure what will happen, but in the hearing phase, it'll probably be epinephrine on trial. Like, how safe is this drug?
Well,
But
I'll
if it
make sure
passes,
that
it'll be
Drew
kind of
knows
hilarious
that.
because it'll sort of completely skip over all the concerns that many people have raised about the drug.
It's supposed to be
Yeah.
pretty
You
minimal.
know, my mom had it.
We'll see.
And I think
We in California actually passed a law last
Did they
year.
pass that?
Yeah, it was like,
I don't
call it
want
allergy
that.
vigilante, so you take a training.
Well, I know I'll
state
have
training,
to,
you get a certification,
I know I'll have an
carry
iPad
your own
shot.
FE device,
Yeah.
and then you're actually allowed
Yeah.
to administer
I don't know
per your
how
training.
long,
But
she
the Good
has
Samaritan is
it
different because it
on,
basically
but,
taps into the existing
it's
infrastructure and all
amazing
these sort of
the
free-floating
recovery is
FE devices.
quick.
Yeah, you could have
Actually,
those
I also
read,
have
you know, like when Carrie
a
Beard, she's
young
talking
friend
about having them
who
in restaurants,
had
you know,
it
like
done, so.
instead of fire extinguisher there, you
Well,
have.
right, that will happen eventually, but this is different. This basically acknowledges that many parents and, like, say, schools and various places have devices around, and that simultaneously you have events on the side. So why not just use that's one more step beyond. Like I carry right now, I carry what an OBQ.
Oh, okay.
It'd be illegal for me to use it. Like, say, if somebody had anaphylaxis in the room or on the street, I can't use it theoretically. I'm going to anyway. See how light it is.
I know, it's just amazing.
Like, this is a real one. I mean,
It's the demo one.
Yeah.
It's incredible.
One of these days, I'm going to get the gall and courage to actually give myself an injection, like during a hearing or something, to show people that it's okay.
Well, you can set up right there or something.
We just did the Allergy Dream Club, and I am having difficulties on the screen here getting the system to shut down. So all I did, normally what works is just to hit the system shutdown button. Yeah, it's really weird. It just, yeah. So I don't know. Should I just hold the the main button down at the bottom? Just hold it for a couple minutes. Will that shut it down, or do you have to actually shut it down by the system shutdown? Because I've hit it probably, I don't and it's still